Hi guys, and welcome to today's lesson. Today we're going to look at oogenesis, so the other half of gametogenesis that we introduced briefly last lesson. Before we do that, we're going to do a quick recap on spermatogenesis so that you've got something to compare it to. Okay, so spermatogenesis begins with spermatogonia, which are stem cells that are inside the testes. So these guys here are stem cells. These stem cells lay dormant in the body until the male reaches puberty, in which there's an influx of hormones that awakens them and pulls them out of dormancy. Once this occurs, they begin to divide. Now, these guys here are stem cells. They contain, so they're normal body cells, which means they are diploid. Now, these guys being diploid cells also means that they have all of the chromosomes, or 2 full sets of chromosomes. When they divide, they create 2 identical daughter cells. So they're dividing to create more spermatogonia. So the process that they divide by is mitosis. Once it divides into 2, one of the spermatogonias returns back into the testes, while the other one goes on to become spermatozoa, or a sperm cell. After this one here has divided and it's now again still a spermatozoa, spermatogonia, and a diploid cell, it then begins the process of meiosis. In the first division of meiosis, DNA is swapped, so we know that stage, the prophase stage in meiosis has a crossing over in which the DNA is swapped between the chromosomes to create variation. And then the chromosomes are pulled apart, the homologous chromosomes are pulled apart into separate cells. So because in meiosis, the homologous chromosomes are the ones that are pulled apart, not the actual chromatids that are pulled apart, we end up with half of the chromosomes in one cell and half of the chromosomes in the other cell. So after meiosis 1, these are no longer identical cells, 1 because they've been through crossing over, and 2 because they now have half the DNA or half the chromosomes in these cells compared to their parent cell. These are now haploid cells, and they're haploid cells that are called secondary spermatocytes. After going through meiosis 1, they have to complete the phase of meiosis, and so they complete meiosis 2 by having their chromatids pulled apart. So once that happens, we have 4 spermatids that are all haploid cells. So 4 spermatids that are haploid cells. We then know that after 6 weeks, so if we add 6 weeks of nurture and development, these haploid cells that are spermatids and form a tail so that they're able to travel through the female body, and they also gather special enzymes at the top of the head of the cell, and these enzymes are there so that they are able to enter the egg cell and fuse with it to break through the barrier of the egg cell, so that the DNA that's on the inside can actually get into the egg. So that's spermatogenesis in a nutshell. Before we go on, I just wanted to clarify what these numbers in the inside of the cells mean. If there is 2n, 2 refers to the number of sets, and n refers to the chromosomes that are in a set. So in this case here, this one is a diploid cell, and therefore there are 2 sets of chromosomes, meaning that there are 2 sets of 23 chromosomes. Remember, each set, 1 set comes from the mother and 1 set comes from the father. Okay, let's have a look at oogenesis. So oogenesis is the production of the female gamete. Gamete or sex cell, same thing. This occurs in the ovaries, and it occurs just like in spermatogenesis, from the stem cells. Stem cells in spermatogenesis are spermatogonia, and in females, the stem cells that creates the female gametes are called oogonia. The female gamete, we're just going to clarify quickly. If we've got one, we have it's creating an ovum. If it's creating many, they're called ova. So this is the other word for an egg cell. So we'll add that in for your page. Okay, so it all begins with stem cells called oogonia. And these guys here form during the early embryonic development and undergo repeated mitotic division to create diploid primary oocytes. So this is a very similar stage to our spermatogenesis process, in which the spermatogonia undergo mitotic division. So they're creating more of exactly the same cell. Stem cells hold all of the DNA, and so therefore all a full and complete set of chromosomes. So when they're going through mitosis, they're producing more full, complete cells with all of the DNA in them. After the diploid cells, so diploid primary oocytes, have been created, there are no further mitotic divisions after birth. So these mitotic divisions continue and continue and continue while in the embryonic stage, but once birth occurs, there are no more mitotic divisions. So in our spermatogenesis process, these divisions that are occurring in the spermatogonia continue and continue and continue. They don't start until puberty, but they continue for the entire life of the male. In females, these mitotic divisions stop at birth. So at birth, the number of oogonia that exist or that have been created are the possible number of eggs, will provide the number of eggs that the female has for her entire life. So a female doesn't have an endless supply of eggs, whereas the male, because their stem cells are continuously dividing, has an infinite number of sperm cells that can be created throughout their life. Okay, so primary oocytes are surrounded by follicle cells, and once this occurs, then meiosis 1 begins and stops at prophase 1. So pre-birth, we'll recap that. Pre-birth, the female has oogonia, which are special stem cells. We'll highlight that here, just to underline it. Special stem cells that divide and divide and divide and divide, and in that time make as many stem cells as they can. Those stem cells then become surrounded by follicle cells, and meiosis 1 begins. But it begins and stops not at the beginning or at the end of the cycle, it stops at prophase 1. So if you recall what prophase 1 includes in meiosis, it includes condensing, and it includes crossing over. So remember, crossing over is where the DNA is swapped between the homologous chromosomes, the one from mum and the one from dad that match. And so the DNA is kind of shuffled around to create more variation. Then the female is born. And not a lot happens, well actually nothing happens in this scenario until she reaches puberty. Once puberty occurs, meiosis continues when the hormones are cyclically released and stimulate only one primary oocyte to complete meiosis 1. So we have all of these cells, the entire number of cells that she's created before birth. So from the embryonic stage and right up until birth, all of these cells have been created. They then get to the point where crossing over has occurred and everything stands still. The baby is born, so the female is born. Nothing occurs until puberty. At puberty, once the menstrual cycle begins, that's that cyclical cycle that we're talking here, the hormones being released in a cycle, they stimulate the one primary oocyte to complete meiosis 1. So only one follicle cell is actually stimulated to continue meiosis. So slight difference with meiosis in this process. Meiosis does divide. So we know that in meiosis 1, the homologous pairs are pulled apart into separate cells, not the chromatids. In this process, they divide, but they don't divide equally. So there ends up being a large haploid secondary oocyte and then a small haploid polar body, which is what we call. The smaller haploid polar body, so this is a key thing too to note, both of them are haploid, so there's still exactly half the amount of genetic information in each cell, but one is just physically smaller than the other one. This smaller polar body usually degenerates, so it usually breaks down into the body, while the larger secondary oocyte goes through to meiosis 2, and where it continues its process right through until it hits metaphase 2. So metaphase 2, remember, is where the chromosomes are lined up in the middle of the cell, and the spindles are attached and they're getting ready to pull away in anaphase. So once they've lined up in the middle, this process stops again. And it stops and it waits for ovulation to occur. So during ovulation, that is when the egg is actually released from the ovary, and the egg travels down, the oocyte travels down down the oviduct. At this point, remember, it's still not completely gone through the whole process yet. At this point, down the oviduct, the egg will either become fertilized or it will not become fertilized. So the next phase is totally dependent on whether there is a sperm cell present or not. If the egg is not fertilized, that oocyte is actually lost with the menstrual cycle. So nothing occurs and it doesn't complete this process. If the egg is fertilized, meiosis 2 is completed, and it produces 2 uneven haploid cells again. So remember, we end up in the last one, we had a large haploid secondary oocyte and a smaller haploid polar body, which then degenerated. The same thing happens again. So meiosis 2 is completed, produces uneven haploids. The larger body becomes the zygote, which is the fertilized egg that will go on to become the baby. And the smaller polar body will degenerate, and it will also be lost or absorbed and broken down by the body. So there are some very key features here that are quite different from the spermatogenesis process. One is the many stoppages throughout the entire process, and two is that whilst one stem cell in the male system turns into 4 sperm cells, one stem cell in the female system actually only turns into one oocyte or egg cell that can be fertilized. So that's another difference. And then of course the other difference is going to be the fact that this process of mitotic divisions, the number of egg cells that a female can possibly make in her lifetime and release, is finite. It is set and it cannot be changed after birth, whereas the male process has a much less limits on its abilities. Once it starts, it can continue for the rest of the life. I'll quickly show you and take you through this diagram here. Basically what we've got at the beginning is our oogonia cell that is produced and goes through mitosis many times. We're looking at about 200,000 primary follicles that are available at birth. Something that's quite surprising though is that usually only about 450 of these follicles develop and go through this process. So up until birth, we've got mitosis occurring non-stop. When birth occurs, not a lot until puberty happens. After puberty, we go through meiosis 1, which occurs in the cycle, so every 28 days or whatever the menstrual cycle is for the female, which then halts again, and the next stage is dependent completely on whether or not there is a sperm cell available. If there is a sperm cell available, meiosis 2 will be complete, and the secondary haploid cell that is created will degenerate, while the sperm cell and the egg cell will fuse and create a zygote, which is why there is 2 sets of chromosomes in that final cell. And that's the summary for oogenesis. Good luck with the questions.